This is part 2 of a lecture on bacterial transformation for April 13th, 2020. And the focus of this lecture is not so much to talk about how bacteria are naturally performing transformation, but how we in the lab have made bacterial cells artificially competent and have kind of taken advantage of this transformation process and exploited it for our own purposes. And so we can actually make bacterial cells that are normally not competent in nature, competent in the lab in several different ways. And one of those is by using a treatment with calcium chloride, or CaCl2. And during a calcium chloride treatment, the positive calcium ions can actually extend pores in the negatively charged membranes of a bacterial cell and keep them open wider and allow DNA to pass through. Additionally, we can use heat or electrical shock to sort of do the same thing and open up pores within the membrane. With heat shock specifically, the heat shock is usually done at 42 degrees Celsius, which causes the lipids in the membrane to become more fluid and be lost, and also results in denaturation of the proteins in the membrane, leaving holes through which DNA can pass and transform bacteria. And usually after a heat shock transformation in the lab, you have to allow your cells a period of recovery to kind of reform those membranes with the now DNA inside of the cell. And the most common sort of competent cell that we make in the lab is E. coli. So why would we even want to make a bacterial cell competent in the lab? The main reason that we do this is so that we can introduce DNA or genes of interest into this bacterial cell and use the bacteria as basically a replication factory to make many, many copies of our DNA or gene of interest. And this process is usually called molecular cloning. And you can see it depicted over here on the right. And so you'll have a plasmid or a circular piece of DNA into which you can insert your DNA or gene of interest from whichever cell that may be. And you can make what's known as a recombinant piece of DNA, or a piece of DNA that combines both plasmid and gene of interest. And then that plasmid can be transformed into a bacterial cell that's been made competent to make a recombinant bacterium. And any DNA molecule that's used in molecular cloning is often known as a cloning vector. And vectors are DNA molecules that can replicate independently outside of the host chromosome, and that they can also maintain the inserted foreign gene or DNA within the host cell and pass it on to subsequent generations or to all of the progeny of the mother cells. And cloning vectors, there are several different types. The most common is a plasmid, but there are also some viral vectors that can be used to introduce DNA into cells through transformation. What all cloning vectors have in common are sort of these three specific features. The first of which is an origin of replication, or a start site for replicating that plasmid. And this just allows your vector or your plasmid to replicate or make more copies of itself inside the host cell. So you can see this cloning vector over here. This is PUC19. It's a very commonly used cloning vector. It's a plasmid, and it has an origin of replication or a sequence that signifies like replication start site, signified here in yellow. And cloning vectors also need to contain what's known as a multiple cloning site, or an MCS, which is just a region of the plasmid that has many different restriction sites or cut sites. And by cutting in this multiple cloning site, it allows you to insert your gene of interest, or that piece of DNA, from a donor cell into the plasmid here in the multiple cloning site so that it can be expressed in the bacterial cells. And cloning vectors also need to contain some type of selection marker so that you know when transformation has been successful. And what a selection marker is just any gene that allows your cell to survive under specific selection conditions. Most commonly, selection markers are antibiotic resistance genes. In PUC19 here, this is an ampicillin resistance gene, so it's resistant to the antibiotic ampicillin. And this will allow you to pick out any bacteria that have been transformed based on their resistance. And so if you grow bacteria containing this plasmid with the ampicillin resistance gene on a plate containing ampicillin, they will grow just fine. Whereas if they weren't transformed with this plasmid successfully, any bacterial cells that are plated on the antibiotic ampicillin plates will die. And so selection markers are used to identify what are called positive transformants. And so how can we tell which bacteria have been transformed? So I've illustrated this idea of using a selection marker out here on this slide. And so during the transformation process, you take a plasmid, right, in the lab or a cloning vector, and you introduce it into competent cells, which are able to be transformed and take up this plasmid. Usually you introduce electric or heat shock to force that plasmid into the competent cells. And this process is not 100% efficient, right? So not every single cell in this pool is going to be transformed effectively. Some of them are going to end up with no plasmid, whereas some of them are going to end up with the plasmid that you're interested in being transformed successfully. And those cells that don't receive this plasmid don't have an antibiotic resistance gene. And therefore, when you plate these cells on media with an antibiotic in it, they won't grow. However, the cells that receive the plasmid or are effectively transformed also receive the antibiotic resistance gene and can grow on a plate that's impregnated with antibiotics. And so you can differentiate between transformed cells and untransformed cells based on whether or not they grow on antibiotic media. This can also be made a little bit more intricate or complicated. And so in some cases, there are additional ways to differentiate between bacteria that can all grow on a plate with an antibiotic. And so in this case, you have a cloning vector here that has an antibiotic resistance gene, and then your gene of interest can be seen down here in blue. And you're trying to insert this gene of interest into the vector. But what you'll notice is that these pieces actually could fit back together pretty easily themselves. And so sometimes you get a plasmid that's re-ligated or basically re-stitched back together that doesn't contain your insert DNA. And some that does contain the insert. And in this case, you can use a special type of vector and differentiate between cells that have your insert or that gene of interest in them and cells that don't by looking for different colors on a plate that's impregnated with antibiotic. And so the cells that have the correct plasmid in them will appear white, and the cells that don't would appear blue. And so not only can you just use antibiotics for a method of selection, you can actually go a step further and also look at color change as well. And so why would we even bother to introduce different types of plasmids into bacterial cells in the lab? There are a couple different reasons why we might want to do this. And the first is because these vectors or plasmids can be used to synthesize proteins of our own interest for different uses. So if we insert a vector containing a gene, that gene can be expressed as a protein, and then we can look and do different things with those proteins. The first is we can produce a lot of them and then isolate them out for commercial use. And so this is what's being done currently with insulin. Insulin used to be isolated from animals, specifically pigs, and pigs used to supply all of the insulin for anyone with diabetes, but we have now harnessed the power of bacteria and introduce vectors that can basically express insulin in E. coli cells or other bacteria. And then we can have this synthetic insulin produced in mass quantities from these bacteria and then sold to treat diabetes. We can also introduce different plasmids containing our genes of interest to study proteins' function and activities, more in like the research lab setting than the commercial one. And so from vectors that are introduced into bacterial cells, we can isolate proteins and study their activities and their functions outside or in test tubes or what's known as in vitro. And we can also, in some cases, introduce vectors that will allow us to see where proteins are localizing and going within the cell and how they actually work in a live organism or in vivo. And so one example of that can be seen here on the right. And there are vectors where you can introduce your gene of interest and then tag it with a fluorescent protein. So a protein that can be excited and that you can visualize in different fluorescent colors. And so you can see a green fluorescent protein here on the left and a red fluorescent protein here on the right. And these are both produced by vectors introduced into Staph aureus bacteria. So these are Staph aureus cells expressing a GFP tagged penicillin binding protein, which will mark the cell wall of the Staph aureus. And these are Staph aureus expressing a M-cherry tagged or red fluorescent FitZ protein. And FitZ is that tubulin homolog that's responsible for making the septum during cell division. So you can see it localizing here in the middle of two dividing cells. And so by creating these fluorescent protein constructs using vectors or plasmids that are introduced into bacteria, we can see where the proteins are in the bacterial cells, and we can infer some stuff about their functions and what they're doing. But what's important to remember is that this does not only apply to looking at proteins in bacteria themselves. We can take these vectors that we make in bacteria and then introduce them into other cells as well. Specifically, one example of that is introducing vectors that make fluorescent proteins involved in mitosis in eukaryotic cells. So here you can see a eukaryotic cell undergoing mitosis. The red fluorescent protein is labeling chromosomes, the green fluorescent protein is labeling microtubules, and all of the spindle apparatus necessary to separate the chromosomes. And so we can introduce, using vectors, proteins that glow or fluoresce into different types of cells other than bacteria as well. And so these vectors not only give us an idea of where stuff is localizing, but also can be used to assess functionality. And so on the right, there's actually a picture that shows a functional reporter, a functional fluorescent output of a neuron firing and how the neurons are all connected to each other. So when this neuron in the middle fires, you can see a bright flash of fluorescence, and then all of those subsequent neurons that are connected will fluoresce as well. And so you can actually not only look just at localization using an expression vector, but you can also look at the function of a protein. And all of this is kind of amazing. And sometimes I think at this point in science, we've seen a lot of fluorescent images, and we've seen GFP being used for a lot of different things. And so it's important to go back and remember how we generate all of these constructs and how important understanding bacteria, as well as transformation and plasmids, really is to understanding how we generate all of these different really cool tools that we now kind of use in almost everyday life.